Okay, so in this video we're going to talk about the scientific process, and in other words, the specific method that people try to use to arrive at answers to questions about things that they've observed in the world around them. I'm going to note that this video might seem somewhat slow and basic, but some of the concepts that I'm going to introduce here are things that a lot of scientists still routinely get wrong in their scientific work. I see it in publications all the time. So it's worth the time now to ensure that you fully understand and can nail this process as I've described it here. And if nothing else, you're going to discover that this process is going to crop up again and again across your time studying science and really across your life. Okay, so to work through this process, we're going to start out with some observations about biological phenomena that people have been tracking for a long time. And so right here, I've got a picture of an amphibian, and this is a type of toad called the golden toad from Costa Rica. You'll see it in color on the Canvas slides. It is actually gold. So this toad first came to people's attention back in the 1980s, and what people noticed was the following. So one year, these toads were very common. You look around, you see them absolutely everywhere. But just a single year later, at the same place and at the same time of year, these toads were nowhere to be found. So they'd vanished. And in fact, as I've noted here, the last time that they were seen was back in 1989. So at that time, that a group of people was noticing this phenomenon with the golden toads, people elsewhere were noticing another thing. A lot of frogs elsewhere were showing up with all kinds of weird deformities. So as you can see here, so for example, here's a frog with more or less normal limbs, but here's a frog that only has 3 out of its 4 legs. Here is a frog that has 6 legs. Here's one with 5, and here's another one with other deformities as well. And so this act of noticing something is really the first step of the scientific process. So it's making an observation about some phenomenon in the world around us. Okay, so we can't stop at an observation though. If I tell you about these phenomena, you might notice that if it piques your interest, you want to take things a step further. So the next step after observations is to come up with a question. And not just any old question. We're going to really break down the steps of the scientific process here, and so what we want to consider is a type of question that's referred to as a causal question. And so some hallmarks of a causal question include that it contains the words how, why, or cause. And so given now that I've told you a little bit about what a causal question is, what I suggest you do next is pause this video and jot down a quick causal question that follows logically from the puzzling observations of frogs having a lot of limb deformities. Okay, and so now here's an example of a causal question that we can ask. So why do frogs have deformed limbs? And I want to note, so one thing to distinguish between here is a causal question is distinct from a descriptive question. So a descriptive question would be something like, how many frogs have deformed limbs? So with a descriptive question, there's a very direct answer to that question, right? So you can go out and count how many frogs have deformed limbs. You can answer this question, then you're done. A causal question, in contrast, is an open-ended question. So it's a question where there are many potential different answers to that question. Okay, and another thing to notice here is that at this stage, I'm not really trying to answer this causal question, I'm just trying to ask the question in this step. Okay? All right, but that does lead us straight away to the next step of the scientific method, which is to go ahead and try to think of some potential answers or explanations to the causal question. So a lot of people were concerned about these frog deformities, and so they came with all kinds of potential explanations. And that step of the process is a really important step. And I'm going to just come up with a couple of ideas here off the top of my head so you can see what it looks like to go through this process of coming up with these explanations. Or in other words, coming up with a set of hypotheses. Okay, so what might be some things that could cause frogs to have deformed limbs? So we know that, for example, sunlight is something that causes cancer in many cases in people, and maybe deformities are somehow related to things that cause cancer. So maybe something about sunlight's involved. And you have to remember, in this period back in the 80s, people were really concerned about a new hole that had opened up in the ozone layer. So a very popular hypothesis at that time was that maybe it had something to do with UV radiation. Another thing that had come up for a lot of people was, at the time, there were a lot of concerns about increased carbon dioxide in the atmosphere. And increased levels of carbon dioxide can cause a shift in acidity in the environment. And so there are also a lot of concerns about acid rain. So that was another hypothesis. So maybe something about the changes in pH in aquatic environments is contributing to frogs having deformed limbs. We could come up with other ideas as well. So maybe it has something to do with frog nutrition during development. So maybe not getting the right combination of nutrients is causing it. Or maybe there's other chemicals in the environment that are contributing to this problem. So one popular hypothesis was that this could be tied to runoff from things like herbicides or pesticides. So here's a set of hypotheses. We could keep going, really, and that's actually something important to notice also, that so this causal question's open-ended. We can come up with a lot of potential explanations or hypotheses to answer that causal question. Okay, so I've come up with a set of ideas here. That's great, but we can't really stop here either. So if I really want to get to the bottom of why so many frogs have deformed limbs, I'm going to have to come up with an experiment or a set of experiments to test my hypotheses. So that's really our next step, is come up with an experiment. Okay, so now let's take a moment here. I've got a pretty extensive list of hypotheses, and I'm not going to be able to test all of these at once. It kind of exhausts me thinking about that. So for the moment, let's just focus on one of these hypotheses. So let's focus on this hypothesis about herbicide or pesticide pollution in water here, okay? So if I want to test this hypothesis about pollution in water, what I need to do is come up with an experiment that tests this hypothesis specifically. And so there's another type of question that I now need to think about here. So if I want to very clearly demonstrate that it's some form of pollution from an herbicide or pesticide that's causing the deformities, how can I do that and show that it's just that thing and not some other factor? So really, when I'm developing an experiment, I need some kind of reference point. And so if I'm going to come up with an experiment to test this hypothesis, I need to have one group of tadpoles that's in water with a pollutant. So let's just say it's in water with an herbicide. So herbicide plus. And then I need to set up another group of tadpoles in water that doesn't contain this pollutant. So I need more tadpoles in an environment that is herbicide negative. Okay? So this structure here that I'm outlining, where I've set up two different groups of tadpoles, is a situation where I'm working with a variable and I'm coming up with specific categories for that variable. And since I am deciding on those categories directly, I'm either going to go in and I'm going to add herbicide, or I'm not going to add herbicide. So in other words, this is something that I'm manipulating. This presence or absence of herbicide is referred to as being an independent variable in my experiment. Right. So just to reiterate, so that's a variable that I'm manipulating. Okay, and then meanwhile, these specific categories that I've come up with here are referred to as treatments or treatment groups. Right, so I've got herbicide present or herbicide absent. And the group that doesn't have the herbicide added also gets a special name because we can kind of think of this as being a baseline or a reference condition. So this particular treatment is referred to as our control treatment. Okay, so also realize if you start thinking about it, you can imagine creating additional treatments here also. So like, what if we had herbicide at strength 1x, or we could also add herbicide at strength 10x, or herbicide at strength 100x. So that would be getting a bit fancy with our experiment. But my point here is that there could be multiple different treatment groups for any independent variable. There's some other components to my experiment also. So if I want to be sure that it's the herbicide itself causing deformities, and I go back to this list of other hypotheses here, well, what about this hypothesis about UV radiation? Isn't it still possible that UV radiation could also contribute to the deformities? I haven't really ruled that out. If I do think that's the case, then I'm going to need to standardize the amount of UV light across my two experiment treatments. So if I'm standardizing, then okay, so if I'm standardizing, then I'm controlling that thing so that it's constant and it's not going to affect the outcome. So any other elements of the experiment that I'm standardizing, those are going to be referred to as controlled variables. Okay, and notice, so a controlled variable is distinct from the controlled treatment, which is just one of the treatment groups within the independent variable here. All right, so that's two major components of my experiment. And I can keep going with what I standardize, right? The more other factors that I can standardize, the more clear the outcome of my experiment is going to be that if herbicide is a contributor, that yes, that is the thing that's causing frogs to have deformed limbs. Okay, so then the third component is kind of the outcome that I'm interested in from the experiment. And so in this case, we could call that something like the percent of deformed frogs. So that outcome is a third type of variable. So in this case, that third type is our dependent variable. Okay, so that's because we're interested, what we're interested in here is the extent to which these deformities depend on exposure to herbicide, right? So independent is what we manipulate. Dependent is, does this outcome depend on whether a tadpole is in the herbicide group or the non-herbicide group? Okay, so at this point we've come up with an experiment. We've outlined our independent variable, some treatment groups, and some important controlled variables, and we've identified our dependent variable. So then the question is, so what comes after this? And if you said, let's do the experiment, you're close, but we're not going to do it just yet. The next thing we need to do to be explicit about the scientific process is to come up with a set of predictions, okay? Or a prediction. So how do we expect this experiment to turn out if indeed it is this herbicide that's causing frog deformities? So what we would predict is that tadpoles raised in water without herbicide, or with herbicide, will have a greater incidence of deformities than tadpoles in water that doesn't have herbicide in it, right? Okay, so now that we've explicitly stated our prediction, we can go ahead and conduct this experiment and get some results. So let's say we use an herbicide called atrazine. So let's see. So we're going to use atrazine. Okay, and we're going to rear our frogs, and let's say we rear 10 frogs in each condition, and what we actually find is that the number of deformed frogs from our control treatment, so atrazine minus, is 0, or 0%, and the number of deformed frogs from the herbicide treatment is, drum roll please, also 0%. Okay, so that's our result. So we have one more step to go here, and that step is to now directly compare our result to our prediction and reach a conclusion. Okay, so in this case, our result here does not match with our prediction that we just generated, right? We predicted that the herbicide, tadpoles experiencing herbicide would have a greater incidence of deformities than those that did not experience herbicide, but as it turns out, nobody experienced any deformities. Okay, so now what I want you to do is to pay very close attention to what I'm going to say about our conclusion here. So for our conclusion, we always want to directly answer this question. Was the hypothesis supported? Okay, now there's a couple ways that you can shake out the answer to this question, right? So the answer could be yes. In this case, probably not. The answer could be no. So in this case, we could say no, the hypothesis was not supported. Or the answer can sometimes be partly supported, okay? And I want to draw another very important distinction here. So when we reach this conclusion, we have not proven definitively, beyond any reasonable doubt, that herbicides are not involved in frog deformities. So when you're in a science classroom, you need to delete the word prove from your vocabulary. And I actually encourage you to write the word prove in large letters and then draw a giant X in it. So we are never ever going to be able to prove anything using this process. And so why am I saying this? So here's something to consider. So what if there's some kind of interaction between an herbicide and one of these other environmental stressors that we just haven't captured in this specific experiment? Or what if we use the wrong dose of herbicide or just the wrong herbicide altogether? And the reality is that we can never completely eliminate these other possibilities. And this concept, it's fairly straightforward, but it can be kind of hard to come to grip with this concept. Because what it means is that no matter what we do, there's always some room left for uncertainty. And I think it's human nature to want certainty. We want to know, is it the herbicides that's causing deformities and causing frogs to disappear? So the next question is, well, how can we get to a point where there is more certainty? And the answer is that we have to go back to an earlier step in this process. And basically, we need to go back, after we've done this, and we need to repeat the process. So we need to come back up to our set of hypotheses here and think about, okay, well, maybe it's one of these other factors. Maybe we need to revisit, redesign an experiment and test this hypothesis again. So my point here is that this scientific method, although I've outlined a linear thought process here, typically it's never completed in a single shot. So go through these steps. Okay, hypothesis is supported. No. Okay, we can go home. No. Instead, we have to go back up to our hypotheses, come up with a new experiment, come up with a new set of predictions, get more results, and see what kind of conclusion we reach the next time. And again. Okay, so it might seem like, I've kind of picked an arbitrary example with these frogs, but I actually picked it for a reason. And that's because there's an undergraduate student researcher who's named Peter Johnson, who got interested in tackling this causal question about frog deformities. And the pesticide hypothesis was a very popular one, by the way, but Johnson actually came up with a different hypothesis. So it's outlined here. That maybe the factor causing the deformities was actually a frog parasite, so a type of trematode called Riberoia. So that's this Riberoia actually has an alternate life cycle. So here's what this parasite actually looks like. It spends part of its life cycle in snails, and then part of its life cycle in frogs. And so his hypothesis is that this could be what's causing the deformities. Okay, so now just to give you another example of an experiment, here is Johnson's experimental design. So he collected a bunch of tadpoles from the field, he set them up in the lab, and in this case, what I want you to notice is that he had 4 different treatment groups for his independent variable, right? So he has a controlled treatment here with no Riberoia, and then he has low, moderate, and high infection levels for his experiment. So that's his independent variable. He had to do the same thing, think about how to control other factors that could affect the outcome of his experiment. So he had to think about controlled variables, and then he had a dependent variable, so the thing that he actually measured at the end of the day. So at this point, I want you to take a moment to think about what Johnson's prediction will be if his hypothesis about trematode parasites is actually true. And I think you should just take a moment to pause the video and jot down Johnson's prediction. Okay? So once you've done that, now here is Johnson's result. So I want you again to pause the video, study this graph, and try to describe this result in words. Okay, so now if you're going to describe this result in words, what I want you to notice is here on the x-axis we've got the different categories that Johnson created for his independent variable. So the number of trematodes or number of Riberoia per tadpole. So this is where Johnson's independent variable is being displayed. And then here on the y-axis we've got the frequency or percentage of abnormal frogs. So here's his dependent variable, right? And this is a standard convention, independent on the x-axis, dependent on the y. Okay, so what we can notice here is when there are no trematodes, there is no abnormality. Whereas with any amount of trematodes here, there were abnormalities, right? Okay, so that is the result. So now I want you to take another moment and jot down what your scientific conclusion would be. So again, pause this, jot down what's the conclusion. Okay, so now that you've done that, here's what I would conclude. So in this case, the hypothesis is supported. That's simple. So now one of the coolest things about Johnson's experiment is that it was such a good test of a hypothesis and such a huge breakthrough for our understanding of amphibian decline that he was actually able to write a scientific paper about it and get that paper published in one of the most prestigious scientific journals, the journal Science. And he did that as an undergraduate. So I just think that's pretty amazing and an incredible example for us. Okay, so are we done? We know the process. Not really. We're going to work through a more complex example during our lecture time together, and what I want to emphasize right now is that basically we're never done going through this process. So for example, what about tests in other species? Here's a test of a more complex but related set of hypotheses in a salamander species, for example. It turns out that in this species, higher levels of herbicide are associated with increased infection with a deadly virus. So for salamanders, in contrast with frogs, the herbicide hypothesis is supported. And then the last thing I want to do is just emphasize one more point. This is crucially important to recognize. And it's that we can never arrive at an absolute truth, but we can use the scientific method to converge on an understanding of how the world works. In spite of not being able to say, okay, we've definitively proven that herbicides cause deformities and have caused amphibian decline, if we repeat this process with a substantial number of experiments across a substantial number of amphibian species, then we can come up with a lot of evidence, maybe an overwhelming amount of evidence, to support the hypothesis that there is a connection between herbicides and amphibian decline. And for all of that, sometimes there can be cases where we think all the evidence is lining up really nicely to support some particular hypothesis about how the world works, and then later on, some experiment comes along that definitively indicates that that hypothesis is actually not supported. And so, what's a scientist to do? So if the evidence is sufficiently convincing, then we change our minds. And I really want to point out that, living as we are in the time of the coronavirus, we've seen it play out in real time over the last 8 months or so. So early on, we started out with a hypothesis about how the coronavirus spreads from one person to the next. So the main hypothesis was that the virus was largely transmitted through droplets, or to use the fancy technical term for droplets, fomites. However, as scientists have continued to conduct experiments and weigh the evidence, there's now evidence that the coronavirus can be transmitted through the air, so it's airborne. So the airborne hypothesis has come to predominate. And this is an example of how the use of the scientific process can have life or death consequences for us and for the people around us. And it can be very confusing to some people when they learn that scientists are this flexible in their thinking, that scientists can change their minds, because it can be uncomfortable to have to deal with so much uncertainty and ambiguity. But scientists change their minds all the time. So to give you another example, there were actually questions for quite a long time about the concept of continental drift, something that we kind of take for granted now. So up until the 1960s, the scientific consensus was that continental drift didn't happen. That's not how the world works. By the 1960s, there's enough accumulated evidence to support the hypothesis of continental drift. So something we take for granted now was definitely not taken for granted earlier. And so again, it can be really uncomfortable to have to deal with the uncertainty and ambiguity of the scientific process. But learning how to deal with that type of uncertainty and ambiguity is an important component of what we are here to accomplish.